Alright, in this video we will talk about the molecules which make up cells. Cells are actually made up of thousands of different molecules, but there are four types of large biopolymers made up of repeating units that make up most of the dry mass of a cell and perform most of the important functions. We'll talk about those one by one. The biopolymers, polysaccharides, which are made up of sugars, membranes, which are made up of fatty acids. Those are the two we'll cover in this video. In the next video clip, we'll talk about proteins and nucleic acids. That would be both RNA and DNA. But before I talk about polysaccharides, I want to say one important thing about the composition of a cell, and that is, whether it's true whether you're talking about bacteria or you and me, is that we are made up primarily of water. Bacteria are made up of about 30% of the dry mass of a cell is about 30%. Now, of that 30%, half of it, 15%, is made up of proteins. Nucleic acids, both DNA and RNA, 7%, are the next largest group. Phospholipids and polysaccharides, each at about 2%. Of course, these are average values, and different cell types will deviate. So these four biopolymers make up about 85% of the cell's dry mass, and it's these four that we will talk about mostly in the first third of the course. Now we'll talk about polysaccharides, which are made up of sugars. Here we see four different views of the same sugar molecule. This will be the most important for this course. This is a molecule of glucose. Glucose is made of 6 carbons, 1, 2, 3, 4, 5, and a sixth carbon above the ring. It's a 6-membered ring because we have 1 oxygen in there. So in the ring, 5 carbons, 1 oxygen. This is called a pyranose ring. The stereochemistry of each of these carbons is very important, whether the hydroxyl group is below or in this case above the ring. Different orientations will be actually different sugars, and they will have different functions inside the cell. This is an example here of showing the stereochemistry. This is the chair conformation. You might remember that that's more stable than the boat conformation. And here is a ball and chain model, and here is a space-filling molecule. This is the most accurate representation of the molecule. However, it's, unless you have a lot of chemical intuition, not as clear-cut in terms of what the bonding is. For instance, you might be hard-pressed to see a 6-membered ring there. In any case, we will talk a little bit more about glucose right now, and the number of carbons helps define our nomenclature for it. Here we see a triose made up of 3 carbons versus a hexose, 1, 2, 3, 4, 5, 6. And these are called aldoses because they have aldehyde groups at the end of them. But we're only showing here the linear conformation. These sugar molecules can isomerize between linear conformations and ring conformations. So the way those will cyclize, in this case, you'll see carbon 1 is right here in the ring. This hydroxyl group on the, the 5th carbon will attack this carbon that has the aldehyde group. This aldehyde oxygen will be converted to a hydroxyl group, and this 6th carbon will be above the ring here. So you have a 6-membered ring, 5 carbons, 1 oxygen. This is the sugar ribose, which is 5 carbons. Same sort of thing. The hydroxyl group on the 4th carbon attacks here at the carbonyl group, and we'll make a 5-membered ring. Ribose, this is the sugar of RNA. If this hydroxyl group was changed to a hydrogen, then we would have DNA. This portion just highlights the stereochemical aspects. If we look at the difference here, the only difference between galactose and glucose is the orientation of the hydroxyl group at this 4th carbon position, but they will be used quite differently. Now, polysaccharides are just complex chains. Here's a linear chain. They can also be branched. We have 4 different glucose molecules connected. This is a starch called amylose. They are connected carbon 1 to carbon 4 by a glycosidic bond. It's called an alpha linkage because the oxygen atom is below the ring structure. We won't talk as much about starch as some of the other biopolymers, but that is what they are made up out of. Now we will talk about fats. Fats are made up of fatty acids, which have long hydrocarbon tails, just carbon and hydrogen. They're called fatty acids because it has a carboxylic acid group here at the end. These are extremely hydrophobic, and these will avoid water at all costs. The fatty acids can be comprised of different types, starting off with a fully saturated carbon. That means there's all single bonds, no double bonds, and it's saturated because it has as much hydrogen on it as possible. Now, when we talk about unsaturated fats, that means there's a double bond somewhere in this chain. The fact that both of the carbons are on the same side, if we looked at this double bond here, both of the chains are on the same side, so this would be a cis fatty acid, and if it were a trans fatty acid, the tail would come down somewhat in this direction. And as you know, there are health concerns with trans fatty acids, and certain municipalities are trying to ban them now. But when we look at the space-filling models, we notice that a real large consequence, geometric consequence of that double bond is the overall shape of the chain. These unsaturation makes it highly kinked, and if you can imagine, we're not going to be able to pack as many fatty acids as closely in a defined space. And so fats that contain unsaturated fats will be less densely packed, and they'll be more liquidy. Like, for instance, if we thought about oils versus butter. Butter is a saturated fat. It's a solid at room temperature because you can pack so many fats together. Now, in addition to fatty acids, when we're building up fats, there's a molecule glycerol, a 3-carbon and with 3 hydroxyl groups, and in this case the glycerol has reacted with 3 of the fatty acid chains to make a triacylglycerol. And as you can see, each the fatty acids need not be the same. We have 2 saturated and 1 unsaturated chain. So that combinatorially there are many different types of triacylglycerols and diacylglycerols in a cell. Now here, when we're building up a phospholipid, which is the type of fat that's in a membrane, we can see the glycerol, the 3-carbon unit. We've got 1 hydroxyl group connected to a fatty acid, a 2nd hydroxyl group connected to a fatty acid, and the 3rd hydroxyl group is connected to a hydrophilic region, a phosphate group, and in this case choline, which is similar to amino acids. And so this is phosphatidylcholine. This is one of the primary constituents of eukaryotic membranes. Again, this region below the glycerol backbone is going to be very hydrophobic. This is going to be in the center of the membranes. This portion, the hydrophilic region, will be facing outwards into the aqueous environment. And lastly on this slide, we'll begin to see how the individual phospholipids are organized to form a membrane. In each case, we have the hydrophilic heads orienting themselves next to the water. The major goal here is to separate the water from the hydrophobic tails. One way of doing this is to form a bilayer where we have 1 layer and then down here a 2nd layer where the fatty acid chains are packed next to each other, and so that water is not on the edge here. We have a 2nd set of phospholipids with the opposite orientation, again, so that the phospholipid groups are near the water. And in this way, the tails are protected from the water. Now this is a cutaway view. We do have problems. The sides of these lipids are exposed, and that cannot exist. We'll see in future lectures about how the cell is going to solve that problem. Okay, in the next video we will talk about proteins and nucleic